Hello everyone, my name is Asma and I am going to present in this poster our method called the Multitask Group Lasso for Genome-Wide Association Studies in admixed population. So let's start first presenting GEOs. Our goal is to find association between the genotype given by single nucleotide polymorphisms, SNPs, and a studied phenotype. In case control design, we focus on comparing the frequencies between a group of healthy people and another group with a specific disease. When these frequencies are different between cases and controls, the corresponding SNPs are therefore associated with the disease. When we do GEOs, we found many problems and challenges. Let's start first by presenting single marker analysis, where we consider only the effect of SNP separately. This is why we decided to move to multivariate approach based on machine learning techniques, more precisely feature selection based on regularization terms, such as Lasso, which presents an L1 norm, and Group Lasso, which presents an L1, L2 norm. Another major problem in GEOs is population stratifications that occurs when we have admixed population within samples, and then we can find association due to differences in ancestry rather than association with the SNPs. To address this issue, we decided to explore multitask frameworks where each subpopulation is considered as an input task. Then we have also linkage disequilibrium that represents also an additional issue where adjacent SNPs on the same chromosomes can be correlated. To deal with that, we used hierarchical clustering to obtain groups of strongly correlated SNPs. So then we can perform feature selection at the LD group level instead of the SNP level. So these multivariate approaches are computationally expensive, when especially they are applied to GEOs data that can include millions of SNPs. We incorporate, gap-safe screening rules that consist on eliminating useless coefficients in the optimization, so then we avoid memory errors and we get significant speed up. Finally, feature selection is also known to lack stability. This is why we decided to include subsampling procedure, which we call here stability selection, in order to improve the stability and to make sure that only consistently selected variables remain on the final selected set. So now we will be able to present our model called Multitask Group Lasso. It is basically designed to assign a task to each subpopulation as an input. Using Group Lasso that is based on L1, L2 norm, we provided feature selection at the LD groups level that we already predefined using hierarchical clustering. L here is the loss function that could be quadratic if the phenotype is quantitative and logistic if the phenotype is qualitative. BG is our matrix of regression coefficients across all tasks for the SNPs of a given LD group. So the goal of our method is to provide selection of LD groups associated with the phenotype across all tasks or subpopulation or specific population, as we can see in the graph here. In order to evaluate our method, we run first simulation with predefined disease loci, so we will be able to detect false positives and validate the efficiency of our model. Then we compared the multitask group Lasso to different scenarios based on the state-of-the-art methods. First, we have the Lasso after PCA adjustment for population stratification, Group Lasso after PCA adjustment for population stratification. So here, PCA adjustment consists on including top principal components as covariate in regression models, and we consider then the residual as a new phenotype, which is qualitative. Then we try to separate the subpopulation and apply a lasso at the SNP level or either group lasso at the LD groups level. So we computed also the stability using the Pearson index stability measure to estimate the variation effect of LD groups along subsets. We computed also the running time for each technique. Now let's present some results. We used basically two data sets. The first one is simulated with 4,000 samples from European and African population, and we included 1 million SNPs. We also predefined the disease loci in different chromosomes, and we computed the LD groups using hierarchical clustering. So on the rock plot here, we can see that our method, multitask group lasso, outperforms the state-of-the-art methods with a low false positive rate. Computationally speaking, our method is slow comparing to the other methods, but this is still reasonable as the multitask group lasso incorporates many components like stability selection with subsampling, hierarchical clustering, group lasso on multiple tasks. In terms of stability, we can see clearly that the multitask group lasso is have the best stability index with 5,623 groups. Then we used drive breast cancer oncoarray data, which includes 28,000 of samples from different populations and around 300,000 SNPs. We can see again that our method outperforms the state-of-the-art techniques with 0.43 as stability index for 62 selected groups. So here I wanted to mention, in order to choose the subpopulation in real data, we used principal component analysis to choose the exact number of tasks. So here, basically, we have 2 tasks. Thank you for your attention.